Hello, everyone, and welcome to the ISAC webinar on monkeypox. This is a topic that has really occupied a lot of people recently after the COVID pandemic relatively stabilized. We had a new virus to really raise awareness and concern around the globe. And for that, the ISAC viral study group has decided to put together this webinar to address various aspects of the monkeypox virus. This program is supposed to be chaired by Professor Fatma Amer, and unfortunately, she's a bit delayed. And for that, I'm going to take over to chair the program until she joins us. Professor Fatma Amer is a professor of medical microbiology and immunology at Zaghazir Faculty of Medicine in Egypt, and she's the chair of the ISAC Viral Infection Working Group and a committee member of the International Society of Infectious Diseases. So without any further delays, I'd like to introduce our first speaker. The way we're going to run the program, we will have 10 minutes for each speaker to deliver their presentation, and then we will leave the questions till the end of the program. So it's now my pleasure to introduce Professor Noha Hammad. She's an associate professor at Zaghazir Faculty of Medicine in Egypt, and she will talk to us on monkeypox, virology, and immunology. Dr. Noha, can you share your screen?
Good morning, good afternoon, and good evening for all our audience from all over the world. First of all, I would like to express my deepest gratitude and thankfulness to Professor Fatma Amer for inviting me to talk about monkeypox virus from virology and immunology points of view. Monkeypox virus is an enveloped double-stranded DNA virus with a linear genome. It belongs to Poxviridae family, which has two subfamilies, including Entomopaux varinae viruses, which infects insects, and another subfamily called Cortopox varinae, which infects vertebrates. Cortopox varinae has identified 12 genera. Monkeypox virus belongs to Orthopoxvirus genus, with other 9 species, some of which are of medical importance to human, including vaccinia virus and variola virus. Taxonomy of poxvirus, as demonstrated in front of you, reveals that monkeypox virus is not a direct descendant or a direct ancestor of variola virus, which is a causative agent of smallpox disease. The reservoir, the definitive reservoir of this virus is still unknown. Monkeypox is a viral zoonosis transmitted to humans through bite or scratch from an infected animal. Human-to-human transmission also occurs through direct contact with infectious sources, scabs, body fluids, respiratory secretions, and intimate contact, including during sex. With the highest risk reported in the current outbreak is among gay, bisexual men who have sex with men. In endemic countries, variation in the virulence of the virus has been observed in isolates from Central Africa, where strains are more virulent and shows the death rate exceeding 10%. Then other strains isolated from Western Africa, where the death rate ranges from 1 to 3%. Likos et al. In their publication, A Tale of Two Clades, revealed or reported that monkeypox viruses fall in 2 distinct genetic clades. One is associated with Western African locales, and the other is associated with Central African and the Congo Basin locales. Phylogenetic analysis predicated on whole genomic alignments of the whole genomic sequences of monkeypox viruses and on constellations of orthologous genes identified in other orthopox viruses, and with using a predicted change of 5 or more amino acids as an indication of potentially altered protein function. Several proteins with differences conserved across the 2 clades were identified. Some of these proteins are involved in various aspects of virus life cycle. Others are involved in immune evasion and determination of host range. However, the potential mediators of enhanced virulence in Congo-Basin strains or Central African strains included proteins targeting the features of mammalian innate and adaptive immunity, including interleukin-1 beta receptor and complement control protein. And as demonstrated in this table, these proteins are not expressed or even absent in West African strains. Since last May, monkeypox cases were reported in an increasing number of different countries and regions, which are not endemic for monkeypox virus. One possible factor influencing this resurgence of monkeypox might be the genetic evolution of the virus through multiple mechanisms, like gene loss, gene gain, and specific interactions with host proteins. Even that changing the epidemiology of monkeypox has been linked to genetic evolution of the virus. However, the repeated circulation of monkeypox in human hosts favors the emergence of new strains and newly adapted human pathogens. All these mechanisms play prominent roles in determining the virus host range and the undergoing sustained human transmission observed in this or in the current outbreak. Analysis of the first 2022 outbreak-related monkeypox virus genome sequence, which included 15 genomic sequences or genomes isolated from several countries, revealed that the multi-country outbreak most likely have single origin of West African clade, which is most closely related to viruses associated with exportation of monkeypox virus from Nigeria in 2019 and 2018, and has been exported to several countries, namely Singapore, Israel, and the United Kingdom. The outbreak virus diverges in a mean of 50 single nucleotide polymorphisms, and this divergence is far than one would more expect, considering the previous estimates of the substitution rate for orthopox viruses, which ranges from 1 to 2 substitutions per site per year. Such divergence might represent a recent evolutionary jump in the current monkeypox virus. Fine inspection of the mutation profile of these 50 single nucleotide polymorphisms further revealed a strong mutational bias, being guanine, adenine to adenine, adenine, and thymine, cytosine to thymine, thymine nucleotide replacements. This mutational signature might suggest the potential action of host ApoPEC3 enzymes, which have host defense or innate immunity to the viral genome editing. The first signs of this microevolution within the current cluster included emergence of 15 single nucleotide polymorphisms, which show the same mutational bias described before. And again, this points to the continuous action of ApoPEC3 enzymes during monkeypox virus evolution. Among the 7 phylogenetic branches directly descendant from the recent ancestor of 2018, A subcluster of two sequences has been observed and identified. And this two sequences in that subcluster share a frame shift mutation or a frame shift deletion mutation, which is present in the gene coding for ankyrin or host range protein. And this gene loss event is hypothesized to correlate with human-to-human transmission identified in this outbreak. What's happening now? It seems that monkeypox virus has entered the population as a sexual foe through opening an ecologic niche and an immunologic niche. Preliminary signs of microevolution coupled with the well-identified fluctuations in the gene content of orthopox viruses can provide opportunities for new establishment of new reservoirs outside Africa and adaptation of monkeypox virus to human hosts. Waning of herd immunity through eradication of smallpox and cessation of smallpox vaccination in the 80s of the last century has been singled out as an important contributor to the emergence of monkeypox in endemic areas. In addition, orthopox viruses exhibit multiple immune evasion strategies to challenge the defenses of a naive host. Last but not least, it seems that the characteristics of the infected population in the current outbreak that will be discussed in depth in later talks contributed to some extent in paving the way for opening both ecologic and immunologic niches that monkeypox virus can exploit. Finally, since monkeypox is closely related to variola virus or that causes smallpox disease, smallpox vaccines can be used for prevention of monkeypox disease with effectiveness of about 85% as reported from past reports from endemic African countries. Finally, small or monkeypox vaccines. There are two types of FDA-approved vaccines: the ECAM 2000, which is a replication-competent vaccinia virus or live vaccines, and the other is the replication-deficient vaccinia virus, which is a live attenuated vaccines. Thank you for your attention.
Thank you. You hear me, please, all? Do you hear me?
Yes, Dr. Fatma.
Yes, okay. Thank you, Dr. Noha, for your presentation. I'm sorry, there is something wrong with my connection. Anyhow, this is what I wanted to say at the beginning. We have started this webinar at the time we are still reeling from the COVID pandemic. Another virus to get to grips with has emerged. This time it is the monkeypox. According to situation update released by the WHO as the 8th of June, 1,285 laboratory confirmed cases and one probable case have been reported from 28 countries in 4 WHO regions where monkeypox is not usual or had not previously been reported. So what's going on? Is it time to worry? Or are we getting overly excited having just lived with COVID? We have gathered today to find proper answers from eminent speakers from different parts of the world. Because of time limitation and certain problems with our connection, we'll first have all presentation and then we'll leave discussion to the end. We had the talk of Dr. Noha Hammad, Associate Professor of Microbiology and Immunology, Zagazig Faculty of Medicine, and the member of the ISAC. Next speaker is Dr. Marimuthu Kalisvar from the National Center for Infectious Disease in Singapore and adjunct professor, National University of Singapore. It's worth mentioning that Dr. Marimuthu co-authored the manuscript describing the first case of monkeypox imported to Singapore. Dr. Marimuthu will talk about transmission dynamics of monkeypox. Please go on, Dr. Kalisvar.
All right, greetings everyone. My name is Kalisvar Marimuthu. I'm from National Center Singapore. Thanks Dr. Fatma for that introduction. I thought covering a transmission dynamics of monkeypox, which doesn't have much data, will be easy in 10 minutes, but it turned out quite difficult, but I will try to keep to 10 minutes. I think I can. So when we talk about transmission dynamics of any virus, that we can talk about two parts of the virus. One is the infection rate of the virus, transmission rate, and number two is the modes of transmission. So I'll start with the transmission rate of the virus. So there have been the transmission of monkeypox hasn't been very high early in the early 1970s and 1980s. But what some of the two meta-analyses have shown that the infection of monkeypox has been increasing over the decades. Like in this meta-analysis, what the investigators found was that both the known clades of monkeypox have been increasing, with the highest number recorded around the 2000 in the current century, which is a bit worrying and makes us think what has changed. Did anything change? If yes, what? And then previously, prior to this, another paper by David Hammad et al., they actually showed similar finding, but their analysis stopped at 1997, where they also included secondary cases from monkeypox, and they found that the secondary attack rate of monkeypox have also been increasing. You can see in this table I extracted from that paper, it showed that it was 7.5 in the 70s, 9 in the 80s, and around 23% secondary attack rate in the 90s. So it is a little bit worrying, and the authors have probably had some premonition about what is coming, and they actually concluded that at the end of this 20th century, we are thus faced with the re-emergence of human monkeypox in an atypical epidemiological pattern, which is exactly what we are seeing now in the 2020. So there were a few factors that have affected the way how monkeypox infection happens and the secondary attack rate of the infection. So, to name some is the increasing smallpox vaccine naive population. Smallpox vaccine have been stopped in most countries around 1980s, late 1970s, early 1980s. So many of the 20 years old, 30 years old at the moment, I received mine, but many of the 30, 40 years old now may not have gotten the vaccine, so make them more susceptible. And there is also somewhat a waning of immunity that has been acquired from smallpox vaccination in the past. And there's increase in global mobility as well, which actually makes it easier for exportation and importation of infectious pathogens. And increased human encroachment into forested area results in primary transmission of infection. So these are some of the factors that results in the number of cases increasing. And from past studies, this doesn't include the current outbreak that is ongoing. Sharing same room and bed, same household, eating or drinking from the same dish have been considered a risk factor for human-to-human transmission. Interestingly, this meta-analysis did not find an association with assisting with toileting or hygiene or laundering clothes. That's a bit unexpected, but that's what they found. So past experience of imported case or cluster management has been quite good all over, especially in Singapore, where we had one case of monkeypox in 2019. And it was an important case of an executive who came for a business meeting, and he actually reported symptoms suggestive of monkeypox. And we had about 22 community contacts, high-risk contacts that were identified. 19 of them were workshop attendees, 4 hotel staff. 14 of them received ACAM 2000 as a post-exposure prophylaxis. 6 declined the prophylaxis and 2 had contraindications. None of the contacts developed the disease. At the same time, hospital contacts, there were 0 hospital contacts who were at high risk because everyone were fully on full PPE when we received this case, because we already were suspecting an infectious pathogen in a traveler. But the situation may not be the same for the 2022 outbreak. As we know, most of the first few cases are actually coming out from the communities, and that's probably what we will see in Singapore and many other countries, and with some secondary transmission being very likely. So that's the infection risk of monkeypox so far, and we can summarize it as that for the current outbreak, previously established risk factors and transmission risks will need to be re-evaluated, and I'm pretty sure it's ongoing now because the current outbreak seems to be significantly different from the ones that we have been reading and seeing in the past. And then when we talk about modes of transmission of monkeypox, surprisingly, there's very limited data available for this virus that has been circulating for decades in our civilization. So that's a bit disappointing. So most of the data that we are actually looking at are inferred from variola or smallpox or vaccinia virus. So, and monkeypox, there is some data, but there is really not much. So when I was thinking, how am I going to present this modes of transmission data, I thought that I will summarize it in the next 3 minutes or so. I'll summarize what is out there without going into the details. But it's quite interesting in a sense. So we have zoonosis as a primary mode of transmission. This is very well established. It has historically proven and is an epidemiological support which is very strong. It's a driver of endemicity and reverse zoonosis from the current outbreak in a non-endemic countries. Any human transmitting to a susceptible animal in a new country and that animal becoming reservoir is a real concern, and this is what most people worry about. Second is direct physical contact or touch-based transmission by direct physical contact. This has very strong epidemiological support. It has a historical success in being a pathway that is well considered, and preventing it seems to be providing success. It is a logical conclusion, meaning that you have infected fluids and scabs on your skin, and touch will likely cause a transmission. That's logical, and it's probably the likely driver of the current outbreak. As we all know, it's mostly resulting from very close physical contact. Environment-related touch-based transmission is also a possibility, and there is an epidemiological support, but seems to be moderate. And it's logical because if an infected person is shedding scabs that are infected, and another person touch it and then self-contaminate, then the likelihood of infection is high. No laboratory support for environment-based transmission so far. There is no one has swabbed a patient area of patient with monkeypox like we have seen in COVID. There is historical success, and intervening in environment as part of a monkeypox transmission prevention is a historical success. Surfaces that we are concerned about includes infectious fluids. Environment we are concerned about includes infectious fluids, contaminating environment, scabs, respiratory droplets. So indirect droplets includes those that actually fall on the, when I say indirect droplets, I'm talking about those that fall on the ground and contaminate the surfaces. And then there is more controversial, more controversial droplet-based transmission or spray of infectious particle. This has been accepted quite well. There is possible epi support, but usually it's related to the direct physical contact that happens around the same time. Logical, because respiratory phase of the virus has a respiratory phase, and spray of droplets can happen during that period. No laboratory support, meaning no one has sampled a infectious patient, and historically well accepted. And then we go on to near-distance aerosol inhalation. So there is a possible epi support because there is animal-to-animal transmission data and animal-to-human transmission data. And there is also a laboratory data supporting viable virus being persistent up to 90 hours. And logical because there is a respiratory phase, and we know droplet and aerosol are a continuum, not really a dichotomy. So it is also as possible as droplet. And historically unsupported, meaning this is not something people bought before this. Long distance aerosol or inhalation is where proper long distance transmission, or both of these can be called airborne as well. This is probably very less likely in monkeypox, but I would like to see data before I accept or disregard an hypothesis. There's no epi support, no laboratory support. Plausible if near distance aerosol is proven, then this is plausible because then in a unventilated room, the virus can possibly travel further. This is historically unsupported. The last but not least is the semen and cervicovaginal fluid-based transmission, which is going higher up in the discussion value during the current outbreak. There's an epi support, suggestive of epi support during the current outbreak, but this needs more investigation. So what I did was I actually came up with a scale of acceptance scale and also evidence scale for this transmission mode, so we know where we are, which one of these is high up in everyone's mind. So for the zoonosis, very clear, the acceptance scale and evidence scale is significant, is high. And for the physical contact is also high. For the environment, it's very well accepted, but the evidence scale is probably suggestive but needs more data. And for the droplet and spray, it's very well accepted, but there is no evidence supporting it so far. But we cannot say just because there's no evidence that's not happening. We need to find evidence. Similarly for aerosol, but aerosol is not well accepted. But there is no evidence, just like the spray of infectious particles. Long distance aerosol is not so, is least accepted and least amount of evidence present, and same for the semen and cervicovaginal fluid. So I would like to complete this talk by telling what some of the required data that we require moving forward for monkeypox, and I hope countries that are seeing it actually generating this data. We need the proof of presence of viral particle on surfaces and extent of contamination. Proof of viability will be a bonus. Proof of presence of viral particle in the air and proof of viability will be a bonus. Duration of viability of viral particles, including scabs on surfaces in the air and in the air, because we need it for personal article care, quarantine period, etc., etc. And incorporation of genomics to prove chain of transmission, human to human and human to air, human. This is difficult to do, but it was done for COVID, so I hope someone does it for monkeypox. Extent of asymptomatic carriage, close to 20% in older studies, but we need to know what's happening in the current outbreak. And possibility of onward transmission of an asymptomatic from an asymptomatic carrier, that should be investigated, and epigenomics analysis to confirm if sexual transmission is possible. These are some of the required data for the present study. With that, I thank you.
We thank Dr. Kalisvar for this very interesting lecture. And now the next speaker is Prof. Souha Kanj. She is Professor of Medicine, American University of Beirut Medical Center, and Duke University Medical Center, North Carolina, USA, and Radboud University, Netherlands. Prof. Souha will explain clinical presentation and management of monkeypox.
Thank you for the invitation to participate in this webinar. So I'll be going over the clinical aspect of monkeypox disease. Let's talk about the incubation period of monkeypox. The incubation period is usually 7 to 14 days, but can range from 5 up to 21 days. And there is association between exposure and clinical manifestations. During the US outbreak in May to July in 2003, the median time window for potential exposure was significantly lower among individuals exposed at home compared to those individuals who were exposed in other settings, such as occupational health, animal care, from 10 up to 1, so quite a big difference. What are the symptoms of monkeypox? The clinical disease is similar to smallpox. The duration of symptoms and the clinical manifestations vary between 2 to 5 weeks. And the illness begins with a prodrome, non-specific. Patients have fever, chills, headache, myalgias, with some lymphadenopathy. And within 1 week of the symptoms, the skin lesions start to appear. Typically, they appear around 3 days, but in some patients up to 1 week until they start to appear. The severity of the skin lesions seems to correlate with whether the patients had received prior smallpox vaccination, with the resultant protection by 85% against monkeypox. So lymphadenopathy is a prominent feature which is not present in smallpox disease, and the lymph node could range from 1 up to 4 centimeters in diameter. They are tender and occasionally they are painful. Most are cervical and submaxillary lymph nodes, and they are reported more in unvaccinated patients for smallpox. What about the mucocutaneous manifestations? There is often inflammation of the pharyngeal, conjunctival, and genital mucosa. And in terms of skin manifestation, the patient tend to have a rash that progresses through characteristics, macular initially, and then after a couple of days, it changed to become vesicular rash, and then pustular rash. And as you see here, the number of days between each progression from macules to papule around 2 days. But for example, from pustules to scabs, it can take up to 7 to 14 days. These are some of the pictures from the Lancet infectious diseases publication from the UK. And as you see, the skin manifestations tend to be very variable from one patient to the other. Now, what are the complications of monkeypox? Obviously, as any skin disease, we can have bacterial superinfection with various bacteria. There could be permanent skin scarring. These tend to be less severe than smallpox, as it's shown in this picture here. Some patients might develop permanent corneal scarring. Also, they tend to be less severe than smallpox. Other patients might develop hyperpigmentation or hypopigmentation at the site of their skin lesions. And some patients might progress to other complications such as pneumonia, sepsis, encephalitis, as well as death in some cases. Now, the mortality of monkeypox is higher among children and young adults, and the course tend to be more severe in patients who are immunocompromised. A systematic review showed that monkeypox has a case fatality rate of 8.7%. And as we've heard, the clades, Central African clades, tend to have a higher case fatality rate of 10%, as opposed to the West African clade, which is one that gives a fatality rate of 3.6%. Now, until June 2, there were no reported deaths from the current outbreak outside of Africa. Some patients might present with an atypical presentation. In the current outbreak, most cases were detected in men who had sex with men and in bisexual men. Cases presented to sexual health clinics. Some had co-infections with other sexually transmitted diseases. So we need to keep that in mind when we are evaluating the patients. The prodromal symptoms might be mild, or some patients present with absolutely no prodromal symptoms, and they can just present with the appearance of the rash as the first indication of monkeypox. In confirmed cases in Massachusetts and Canada, the rash started in the genital and the perianal region before spreading to the extremities. And in some cases, the rash was limited to one body site. So there's no rule about monkeypox needing to give us a disseminated rash. There are also reports of some patients presented with proctitis or inflammation of the rectum. These are pictures from the current outbreak from patients seen in Italy. And as you see, some of them had anal lesions that progressed over the days. Others had genital lesions, penile lesions, and others with skin lesions that have evolved as I have described previously. Now, what is the treatment of monkeypox? We don't have a lot of data on treatment, but we know that brincidofovir, which is a prodrug of sitofovir, and another antiviral drug called tecovirimat, they are two orally bioavailable drugs that are approved in the US for the treatment of smallpox. They demonstrated efficacy against orthopox viruses in animal models, but they were never assessed in human clinical trials. In a case series of 7 patients with monkeypox from the UK, starting in 2018, brincidofovir and tocovirimat resulted in transient reduction of the monkeypox virus PCR cycle thresholds, as you see in these 2 graphs. But these improvements were not durable or consistent between the patients. As you see, we had virus PCR spiking again. And the CDC holds an expanded access protocol, or in other terms, what we call compassionate use protocols, that allows to use of stockpile drug to treat monkeypox during an outbreak. And they have a program for tecovirimat, one for sitofovir. They also have a program for vaccinia immune globulin intravenous administration, which had been treated and has been used in the past for the treatment of complications due to vaccinia vaccination. For brincidofovir, it's approved by FDA. It was approved in June 2021 for the treatment of human smallpox, not only in adult, but also in pediatric patients and neonates. And CDC is currently developing a program to help facilitate its use as treatment. We've heard about the protection that the smallpox vaccine can give against monkeypox, and I think we need to keep that in mind. And let's talk just over the next couple of slides on the lessons learned from previous outbreaks. We've heard that there are 2 types of exposure. There is the complex exposure, which is invasive, such as a bite, scratch, breaking the skin barrier. And there are other types of exposure, which we call non-invasive exposure. That is, for example, when you have a contact with an infected animal, not causing a break in the skin. There are, you see on this table on the right side, there was no statistically significant association with fever intensity or rash between the complex and the non-invasive exposure. However, individuals with complex exposure were more likely to have pronounced systemic symptoms and to be hospitalized, and this was statistically significant. And also, if we compare the presentation of the complex exposure to the non-invasive exposure, as we see more gastrointestinal presentations in the complex versus more upper respiratory tract symptoms in the non-invasive. And as well, if we compare the incubation period, it tends to be longer in the non-invasive. The rash tends to be also longer duration as opposed to the patient with complex exposure. The UK cases of monkeypox, none of the identified 7 patients had oral lesions, and most experienced a relatively mild course of illness consistent with infection by the West African clade. I think to stay with my allocated time, come to my conclusion. I think monkeypox is mostly a clinical diagnosis. The virus might have been spreading for a while. We have to keep in mind the unusual clinical presentation and a high index of suspicion for atypical presentation. Genomic surveillance is very crucial, and more research is needed to investigate the mode of transmission and reservoirs that might contribute to the current presentation. I stop here and thank you for your attendance.
We thank Prof. Souha for the very interesting presentation. Now the next speaker, we have the pleasure to have Dr. Alfonso Rodriquez, and accept my apology if I am pronouncing the name in a wrong way. Anyhow, is a faculty of the Autonomous University Foundation of the Americas in Colombia and of the Scientific University of the South in Peru. He is currently the president of the Colombian Association of Infectious Disease and vice president of the Latin American Society for Travel Medicine, and editor-in-chief of the Journal of Travel Medicine and Infectious Disease. He is fellow of the International Society of the Antimicrobial Chemotherapy. Dr. Alfonso will talk about the ongoing epidemiology of monkeypox. Please go.
Thank you very much. Thank you very much, Dr. Fatma, and thanks for the invitation. I will discuss these following epidemiological aspects regarding monkeypox. I will talk a little bit on epidemiology, previous epidemiology in Africa, about the global spreading, epidemiology by regions and selected territories, some about the epidemiological variables, and finally some epidemiological considerations for surveillance. First of all, we have to remember, as has been discussed, this is a zoonotic virus. This is a virus that belongs to a group that possibility that infects, in fact, different animals. And this is important from the ecological point of view for zoonotic transmission, in the way that this is influenced, as many other zoonoses, by different environmental, climatic, social, and different aspects that have been, in fact, studied over the time in different African countries. In the past, particularly, as has been mentioned, this was the ecology of this disease where, for example, the transmission was related to the contact and consumption of raw animal food. But right now, as has been explained, we have concerns of different ways of transmission. And this, as was already mentioned, is a matter of concern, including the possibly short-range aerosol that is right now under study. So the question, as has been mentioned in news even, is if it's a time to worry or one to ignore, as has been also mentioned by the director of the WHO yesterday regarding the convening of the emergency committee under the international health regulations that is assessing or will assess if this outbreak represents a public health emergency of international concern, as was initially the case of COVID-19, and in the past, many other outbreaks. We have to remember, for example, Zika in 2016. So previously, the epidemiology is well related to what's already mentioned. In fact, those countries in West Africa affected by the West African clade, with the exception of Cameroon that has both clades, and the situation with Central African or Congo Basin clade, where are the most reported cases in the past. As we can see here from this systematic review from Beer, analyzing the epidemiology in these countries, particularly the case of the Democratic Republic of Congo, but also the Republic of Congo and the case of Nigeria had been very important. And in fact, in the Democratic Republic of Congo, as reported in this paper, has been an increase over this period with the largest number of cumulated cases. So certainly right now with this situation, some authors of this group by Heider and colleagues are highlighting the underestimation of disease importance in different ways, probably related with problems of surveillance or to try to assess the real burden of monkeypox. But certainly the population has crossed a threshold of sustainable human-human transmission, probably not only in the current outbreak, but particularly in Africa. And in fact, a new estimation of basic and effective reproduction rate is right now necessary. But as you may see here, probably in some endemic countries, there are on the recording of cases. But other studies suggest also what was initially mentioned in the first presentation, the impact of the stop of the smallpox vaccination, especially in African countries, but precisely with a difference in the ecological variables, such as the difference in the incidence, but according different ecological areas, for example, in the Democratic Republic of Congo, but certainly with an increase in the average annual incidence per 10,000 population during 2006 to 2007 compared to previous years in different age groups, especially those younger, as you see here, a significant increase. From 2003, as has been mentioned, and go on, imported cases have been presented in different countries. And probably one of the most important outbreaks outside Africa was precisely the 2003 outbreak in United States, which was particularly only zoonotic transmission. There was no demonstration of sustained human to human transmission. And this outbreak, remember us, the importance from the epidemiological point of view of the problem related to illegal wildlife trade, which is the gateway to zoonotic infectious diseases, as was, for example, the case of SARS-CoV-2 and many other coronaviruses. But right now, probably this situation for many other viral infectious diseases. Right now, the concern began, especially in United Kingdom, after this first case confirmed on May 6th. And you see here the increase in the reporting of cases over more than 30 countries in the world outside Africa, mainly in Europe, but also in North America, in Oceania, in Middle East, and even here in Latin America, where I am. And this is also a travel-related problem, but within the 7 days rolling average cases that have been reported over these couple months. And right now, according to source, we have over 1,600 confirmed cases in different parts of the world. This is for today, the most updated, 1,800 cases reported right now in more than 30 countries, according the different epidemiological reports you can see. In this way, it's important also to see what is happening in territories and in different regions. This is particularly most important right now in terms of the numbers and the countries in Europe that has been progressively reporting cases in different countries. This is one of the maps of the European CDC, most updated. And you will see, for example, the importance in countries, for example, such as Portugal and Spain, but also particularly in United Kingdom, the country right now with the highest number of confirmed cases. This is a report from the United Kingdom that shows that most cases have been confirmed in England and particularly in London. And it seems that the number of reports probably decreased after the May 22. In North America has been also important in the United States, but also particularly in Canada with over 124 cases that have been confirmed. And as you will see here in United States, multiple states have been confirmed at least one case, but the case of New York and California are the states with the highest number of reported cases. Here in Latin America, we have also 4 countries that right now have confirmed cases in the region: Mexico, Venezuela, Brazil, and Argentina. In the Middle East, United Arab Emirates and Israel. In Oceania, Australia with 8 cases. But also we have to remember, and has been mentioned, that in Africa also is still reporting cases. And there are 44 confirmed cases from this year, 1,400 suspected cases, but 66 reported deaths so far. What about some epidemiological variables? Well, this has been occurring mostly in males, in sexually active age population. People is presenting particularly with genital ulcers, as you may see from this data from this search. This is a very interesting web collecting data right now. And a few of them, at least 64, have been hospitalized. Most of these have been in Germany, Italy, and Spain. But right now, there is an investigation of a possible death in also in Brazil. Finally, from this publication of the Journal of Medical Virology, you may see here, which is consistent of what we mentioned with the most prevalent signs and symptoms. And cervical lymphadenopathy. Lymphadenopathy, for example, is important and prevalent in these cases. Finally, some epidemiological final considerations. It's important to remember the natural history of disease that may present right now outside Africa in a typical way, to assess the risk and to follow the developed algorithms that right now has been developed by national and international authorities. And I would like to highlight that in different regions, right now, the surveillance is in case to detect probably unsuspected cases that has been reported also in the literature. I would like to recommend this publication from WHO with different algorithms, and I finish this couple final slides to invite you to submit papers to our special issue on human monkeypox, travel medicine, and infectious disease. Thank you very much.
Thank Dr. Alfonso for this very interesting presentation. Now the next and the last speaker is Rehab Tash, Dr. Rehab Tash, Associate Professor, Microbiology and Immunology, Department of the Faculty of Medicine, Egypt. She is a certified associate trainer and certified international professional trainer. Dr. Rehab will explain infection prevention and control measures related to monkeypox. Please, Dr. Rehab. Go on.
Greeting everyone, attendants and dear speakers. Thank you, Professor Fatma, for inviting me to this webinar. In the next 10 minutes, we will discuss the infection prevention control measures for monkeypox. During this talk, we will hit the primary preventive measure for monkeypox as a zoonotic disease. What are the infection prevention control measures to prevent human-to-human transmission in different settings, healthcare facilities, community, laboratory, ecological studies field? In fact, infection control play a key role in outbreak prevention, which is the current situation now. We should, as an infection control preventionist, raising awareness about the monkeypox, fighting rumors and misinformation, and we will see how we be a part of multidisciplinary One Health initiative. Back to basics, it's a zoonotic disease, so prevent the transmission from animal to animal and take measures to prevent the transmission from human to human. In fact, not only animal workers, veterinarians, zoo attendants is responsible for preventing primary transmission. It's the rule of everyone to avoid contact with rodent and the primates, avoid inadequately cooked meat. Control of rodents is mandatory. Animal movement control, especially from endemic areas to non-endemic areas, which begins as a current outbreak. Surveillance and the control of monkeypox in animals, and implementation of biosafety principle in animal holding facility. And lastly, risk communication and public awareness play a key role in primary transmission prevention. Also, for patient confirmed or suspected cases of monkeypox, we should exclude pets from the direct patient environment. So it's a time now for One Health initiative to come to the scene. It's a multidisciplinary, multi-sectoral approach. By year 2019, there was a very flexible, comprehensive, informative guide to put this One Health into action, and it's time now to do so. Now, the infection prevention control strategy to prevent the secondary infection from human to human. Isolation precaution is the base, standard precaution for every suspected and confirmed case. And now the transmission-based precaution is mandatory. Contact and droplet precaution now established to be the basic for managing the monkeypox cases. However, still as previous talks discussed, airborne transmission is about questionnaire and not settled for monkeypox. So do we have to implement airborne precautions or shouldn't we? In fact, extra precautionary, some countries implement full airborne precaution. However, WHO says that just take the respirator in 95, for example, adding them to droplet and the contact precaution, and that's enough. However, full air precaution is mandatory if you suspect a varicella zoster infection until excluded. Also, if the healthcare worker is going to perform an aerosol generating procedure, it's time for airborne precaution fully implementation. How long isolation precaution to be implemented? Until all lesions have resolved and flushed air. However, clinicians should be aware of that severe cases may have a viral prolonged shedding, and each individual case must be assessed accordingly. Infection control measures at the facility level depends on the risk assessment. We have a message for all healthcare workers: please, monkeypox as an documented healthcare acquired infection in the past, both in the endemic and non-endemic areas. So be careful. Anyone in the current outbreak presented to your healthcare facility with rash or fever with a lymphadenopathy, screening and triage is mandatory. Not only that, at the screening area, you should implement the basic infection prevention and control measures. Supply alcohol hand rub, make plenty of masks, keep the distance, avoid crowdedness, booster hand hygiene and respiratory etiquette signs, and any person or patient in the attending area of the screening with respiratory symptoms should wear a mask. Our front defenders, healthcare worker, if you have exposed to a monkeypox case, confirmed or suspected accidentally, please, for every care facility, you should have a risk management plan in place, ready to respond to your healthcare worker inquiries. I have exposed to monkeypox. What should I do? The first question to answer, did you wear your PPE or not? If you have been wearing PPE and implementing droplet and contact precautions, so there is no risk of infection. Otherwise, if you didn't wear your PPE or you have direct contact, you have to consider active monitoring for 21 days, and then exclusion from work for 21 days. Travel is must be excluded for this monitoring period, and vaccination, post-exposure vaccination is recommended. If you have to transport a monkeypox patient, confirmed or suspected case, it's preferable to use ambulance. You will have the infection prevention standard precaution easily implemented. Avoid transporting in public or private vehicles. Driver should wear PPE. The accompanying person should wear PPE. The patient itself should cover all skin lesions wearing a mask, and after leaving the ambulance or the transport, disinfection with 1% hypochlorite is mandatory before the next patient. Every facility having a lab dealing with monkeypox specimen should implement core requirement for biosafety. All healthcare worker lab surface that dealing with monkeypox specimen should have the pre-exposure vaccination according to their national guidelines. To decrease the burden for the healthcare services which have been suffered during COVID, any case who can be isolated at home, send home. However, this decision must be taken as case-by-case decision. How should I take my decision? please refer to WHO checklist for assessment. It's okay to send your patient or not. Now, rumors about the sexual transmission of the current outbreak. It's a precautionary measure by WHO released in 10 June 2020 to have certain infection prevention control for sexually active population, abstain from sex while symptomatic, including the prodromal phase if present, and abstain till all skin lesions are healed and new skin, like isolation precaution. However, please, healthcare workers, separate stigma from health advice. Communicate effectively, and that's how we can't lose the positive recording of cases. If we stigmatize, we will lose the case reporting. So separate stigma from health advice and communicate effectively. Sexually active population should know that condom used for 12 weeks after infection, not for prevention of monkeypox, it's prevention for sexual transmitted disease, but alone can't prevent monkeypox. And as our colleagues and the previous speakers talk about vaccine is a very effective preventive measure, pre-exposure and post-exposure. Thank you.
Thank you, Dr. Rehab, for your comprehensive presentation. Now, because of time limit, we'll have only a couple of questions, please. A question from Izatost Sar. So, since aerosol transmission is possible for monkeypox and COVID-19, can also be transmitted through respiratory droplet, is there a chance of a DNA-RNA hybrid virus, especially in those that may present with the monkeypox COVID comorbidity? The question is to Dr. Kalisvar, please. Dr. Kalisvar is here or he left? Okay. So this is virology. Dr. Noha Hammad is here or she left also?
Yes, Dr. Fatma, I'm here.
Yes. Since aerosol transmission is possible for monkeypox and COVID-19, can also be transmitted through respiratory droplets, is there a chance of a DNA-RNA hybrid virus, especially in those that may present with the monkeypox COVID comorbidity?
We can tell until the whole genome sequence is fully identified for apparent or newly originating or evoluted genes to be identified in the monkeypox genome. However, being monkeypox viruses is larger viruses that replicate in the cytoplasm of the human cell or eukaryotic cells, there are possible routes of horizontal gene transfer between genomes, even the mRNA through reverse transcription and integration of this reverse transcripted cDNA of an RNA virus can be introduced into the genome of DNA or monkeypox virus.
Okay, another question from Vivian Montblouis. Given the observed deletions in West African clades as compared to Congo Basin clade, can one speculate differences in histological tropism as well? The concern I have is that speculate whether virus might accumulate more in saliva with the West African clade. The question is to Professor Alfonso, please.
Well, I would like first of all to complement regarding the previous question. Certainly, there has been no observed cases so far from co-infection between monkeypox virus and SARS-CoV-2 or COVID-19. But what is important to consider and to remember, not only for differential diagnosis, but the possibilities of co-infection is with other sexually transmitted infections. And this was the case of HIV, but also syphilis and others, and this is important. Regarding this question, I think it's early to answer that, and certainly it's important to understand right now the difference between not only the clinical phenotype, but the tropism of the Congo and the West African clades. And it's important from animal studies, and we have been looking on that in certain way, this is a multisystemic virus. In animal studies, you may see that this virus may reach practically any organ or system, brain, lungs, and even testes, different other areas, skins. So it's a multisystemic. And right now in humans, we have to look also on that.
Okay, Dr. Alfonso, you want to add something, if it's possible, in 1 minute, please? Can you? Yes,
well,
go on, please.
Yes, I ask you to add for the previous question. I already mentioned it.
Okay. Okay, that's very good. Now we had, I think now we may find answers, some answers to the question, is monkeypox a new human threat? In fact, many studies are still needed to answer the rest of the question. However, it is our responsibility to do so. Monkeypox is a known virus rather than a new one. We already have vaccine and possible means of treatment. It's mostly mild, although it can be more dangerous in young children, pregnant women, and people with weak immune system. It spreads more slowly than COVID, and the distinctive and painful rash makes it harder to miss than a cough that could be anything. This makes the job of finding people who may have been infected and vaccinating those at risk of catching the infection. Ways of transmission and IPC measures has been illustrated. And let us say an ounce of prevention is better than a pound of cure. Finally, people of Africa need to send a message to the global North media. Through social media, they asked journalists and editors of global North media outlets not to resist and stigmatizing in their monkeypox reporting. Media coverage of monkeypox paints it as an African virus. Monkeypox has been reported in nations worldwide, but a search shows only photos of black. I like to thank Prof. Souha because she presented photos not only of black, but also of white. Thanks to ISAC and Delta Medical, thanks also are due to our distinguished speakers and respected attendees. Thank you and bye. Goodbye. Thank you.